Thank you. Fiona, thank you for the invitation. I'm going to, I would say, switch gears and go into the neuroscience mode. My laboratory has been interested mostly in plasticity. And the question is here. This is a macaque brain, and we see that almost 1/3 of that brain is devoted to vision. And the question is, what happens to these areas where you are deprived for vision since birth? And the mode is here from postmodern plasticity. And by definition, this happens when one sensory modality links to organization in brain representation of other sensory modalities. So there is a switch. And we can see what we were discussing about conjunctive blindness in humans, and where visual cortex becomes either somatosensory or driven by this impulse. Before I go into the human work, I want to show you a piece of animal studies where we reorganize the brain. So this is studies done on hamsters, and the reason is that these animals are born premature, and we can do lesions of their system and show some kind of reorganization. And the normal visual system I'm showing you projections to area V1, the second projection is colliculus to V2, and the third projection from the pretectum to Vn. And if you do some lesions, like, for example, ablating the superior colliculus and cutting the brachia or the inferior colliculus to make room in the midget geniculate, what happens is that you can force projections from the retina to go to the auditory cortex about the MGM. So we have lesions, everything that is on the cortices and the superior colliculus, and end up with something like this. So now you have an animal that has the retinal ganglion cells projecting to the auditory thalamus, which is the medial geniculate, to the auditory cortex. If you want to prove this, you can make an intraocular injection of a tracer, an anterobright tracer that will carry to the medial geniculate that we found the terminals as in here. So this medial geniculate is full of connection of terminals coming from the retina. So now we have an animal that has retinofugal projection directly to the medial geniculate, which is again the auditory thalamic nucleus. Question, of course, does it work? So we put electrodes in the auditory cortex, and we presented to the animal flashes. Then you see a visual response. And the same cell, we presented the various sounds, and you could see that for each cell there is a response. So this is what we call a bimodal cells, and most of the cells in the auditory cortex become bimodal. We looked at orientation preferences, and you see also that in these groups. So you see also that normal animals have orientation preferences for vertical, oblique, and horizontal, and we find the same thing in the rewired animals. And if we look at direction selectivity, we find cells that are also unidirectional and bidirectional. For behavior that is more interesting, we devised a task whereby hamsters were trained in tasks that are very much dependent on the integrity of the visual cortex, which is well known for the past 40 years. And we trained hamsters in a task to choose this combination box. And what we show here is that a group of intact animals you have no problem in fact learning the task. If we add a visual cortex lesion to these animals, they keep the light-dark discrimination, which is what's always been found, could be mediated by so many other structures, but the grading is square, so shapes out disappear. And these animals are cortically blind if you remove the visual cortex. If you do an auditory cortex lesion, they see one, but they are deaf. Interestingly, if we look at the rewired animals here, before, and now these animals have the retinal projection into the auditory cortex, right? So if we test them in the three tasks, as here in black, they do these discriminations very well. And if you lesion the auditory cortex, they become cortically blind also, meaning that in fact the system here has acquired some properties of the visual cortex. So the conclusion is here that we found responsive neurons in the auditory cortex of these rewired hamsters that are similar than those that you see in the normal animals, the visual cortex of normal. If you ablate the auditory cortex in these rewired animals, we have visual protein discrimination deficits similar to those induced by ablation of the visual cortex. And these novel surgically induced retinal projection to auditory cortex are functional at the behavioral and neurophysiological level. And this is really a nice case of cross-normal plasticity where you can really substitute cortices and hence function. So when we published this work, I got a call from Paul Clinic at the time where he was extremely interested with his TDU system and wanted us to do something with it at the neurophysiological level. So now we see how we can turn visual cortex into synesthetic cortex. So I'm going to go over these things quite fast because we have not talked during this past 2 days about these other forms of invasive methodologies. But I'm sure everybody is aware about the domain system where you can implant a grid of electrodes into the visual cortex. The Humayun grid for the retina implant. And I think something that is becoming extremely interesting is a stem cell transplant. And maybe at one point, all these, of course, systems could be of great use for somebody who has already experienced vision. Because all the subjects I'm going to talk about are congenitally blind. And it would be nice to discuss a little later about late blindness. But most of the systems are quite good for late blinds and not really for congenitally blind people. So we talked about this monitor substitution and the tactile vision that we're going to use. Paul and I remember having seen this system before. It's kind of, you can imagine, very bulky. And somebody has already presented in the beginning of this symposium what Paul had produced as data. And the interest was now to look at the systems and their evolution. And we have been using this machine that you will see tomorrow, and you can play with it. And the newer system, the V100, which has been approved by the FDA, the equivalent of the European FDA, so it's on the market in Europe. People can buy it. We can discuss also the use of these machines. The interest with cloud system at the time when Ron Coopers and myself started with it was that nobody had ever used it. The problem working with blind people who have used Braille, when you get them to do the studies, they have been extremely trained because they have started so early, and then it would be difficult to know how the brain was before they started doing subjects, even us, I mean, we've never seen or experienced a machine before. And I believe very much in training induced plasticity, and that's what we're talking about. So what we did, we got the subjects, and at the time fMRI was not readily available, so we used PET, and we did a familiarization period where we showed these codes. We let the blinds feel the machine, explain to them what it is, and then we let them put the grid on the tongue and having what I call the bubble, the champagne bubble phenomenon. And after that, we gave them a training session for a few days. And after they mastered the task, which is here, we did another PET. So the task was an orientation discrimination task, which is a feature, as I showed in the beginning, that is a very strong feature of the visual cortex. So we thought that maybe orientation being one of these primary features, we should start with doing this kind of training. So subject has to respond with their finger up towards the tip of the tongue to the sides and toward the back of the tongue. And this is a student, so the stimuli were represented and the person was lying in the PET scan. So the subjects, as here, most of the subjects we get retinopathy of prematurity. They are over-oxygenated as premature babies at the lose sight. And from liver, and we occasionally find other glands like cancer of the retinoblastomas and things like that. But most of our subjects are retinopathy of prematurity. We have a 7-year-old here. Most of the other studies we've been using where we taken people who are congenitally blind, no visual experience, so they have to be from so we can discuss that also. Why the tongue? The tongue has a first of all, it's in aqueous milieu, so it's a good conductance, and also it has a very large representation into the somatosensory cortex. And this study was Picard and Olivier and I quite a long time ago, and they do it with the Penfield technique, where they were simulating the cortex and asking the subjects where they had the feeling, the sensation, and then they were putting these little flags, as you might remember, and they might this way. We are trying very hard to try and map it bottom up instead of top down, and it's not very complicated. So something we can discuss again later on if you have choose. Our exclusion criteria, of course, are there. And the section you can see here, they actually in the task to criteria, the monitoring back and the control zone. It's amazing because anybody can use the TTU, can be trained in it. Even our blindfolded controls can use the system very easily. So it's easy to know. And if you look at the reaction time, you see also that was training the if we look at the PET before and after training, we see that after training in the blind, they start recruiting the visual cortex, and not the control subjects. And that's reminiscent a bit of the thing that has been done on braille readers initially, I think 1996 or 97, where a braille reader inside a PET scanner activates the visual cortex. The same as areas as people. And also, if we look at work that has been done, visual orientation discrimination by Shikata et al. In 2000, and if you look at the coordinates of these areas, they are similar. So the blind, they activate areas that are similar to people who have vision and are doing an orientation discrimination task. So if the visual cortex is activated or recruited in the blind with training, that means that the visual cortex is there and is functional. So what about the streams coming out of this cortex? So we saw somebody has shown this, and I love this pitch. And the two streams are there, the dorsal stream mainly for motion and the ventral stream mainly for the recognition of shapes. And we did a task, we did a task here where we showed the correct movements, so all the dots were moving in one direction, and left and right, and incoherent motion. So we trained the subjects first, and we show here that post-group also can do the task very well. And when we put them in the scanner, this is flat map of the cortex. What we did simply, what the software did simply is open the fissures and flatten the cortex. And we can have these blocks here of these areas like the visual cortex, the pericardial fissure, the lateral occipital, and also area MT, as you all know, that is the motion area. And we see also, we're surprised to see that tactile motion can also be activated in a normal subject. But I draw your attention here that we did no visual cortex activation in the control subjects when they are stimulated tactile. If we look at these different motion controls, as you saw in the text, you have the recruitment of area MT. So, and again, which is considered in normal seeing people as a visual motion. Then we looked at shapes. This is the shapes that we use. Subjects were trained to recognize these shapes, and we see here that they do that quite well in the performance curve. And if we look at the reaction times, we can see also that there is a decrease in reaction time with learning. If these subjects are put into the scanner, we see the activation of the parts of the ventral stream that is also involved in the recognition of shapes. So the first conclusions would be that with sufficient training, subject can learn to use the tongue to perform simple visual tasks because these tasks are simple. And so the tongue might act as a portal to convey information to the visual cortex in the brain. We see later on how it is possible that your tongue gets connected to your visual cortex. Discourse model plasticity, and it was surprising, can develop really fast. As I showed you in these curves, it's only a few sessions, 5-6 sessions for 1 hour, and then you can start recruiting the cortex. But before training, they never do a show activation of the visual cortex. So it is really training dependent. And what is really interesting is this, that the functional specification of the word cortex into the two streams, the ventral and dorsal, seems to be preserved in the blind as revealed by tensor, the diffusion tensor imaging. So this is a technique where you can see tracts. So we could discuss that also later, and it looks like this. So you have three types of tracts. The inferior longitudinal fasciculus and the frontooccipital fasciculus are part of the ventral stream, and the superior longitudinal fasciculus is part of the dorsal stream. So you have the controls. You have an horizontal view and a mid-sagittal view. to give you an idea of the tracks, and you have the controls, and you have the lines. And what you see here is that those tracks are preserved in the blind. So this cortex, or this so-called visual cortex, I hate to call it like this, but it's the back of the brain. So put the quotes on visual for this cortex. The visual cortex has the two streams, and of course they are served by these fasciculi that are still present. There is a big debate actually if these fasciculi in the blind are enhanced or smaller or whatever. Until we resolve this, they're there, and they are functions as you have seen before. Of course, the inspection, the vision might say that maybe in the blind it's a little smaller or it's a little thicker here, etc. So we're doing all these volumes. It's very difficult to get big samples of congenitally blind individuals. Retinopathy of prematurity now is less and less present because we know why. So we don't over-oxygenate babies in premature babies. So what about navigation? So the company Vicam has put together this system that you will be able to see tomorrow. It will work, but to show you that the way it works, so the camera will take an image and send it onto now the blind person is scanning with the camera wearing the forehead that you see on the other thing, and this is what the tongue is scanning here. So subjects move, scans the thing, and gets the image on these pixels that you see here on the tongue. And the end result is this. And the subjects trained will say that they will recognize, so to speak, the quote-unquote visual streams, because it is the tactile sense. So we built this corridor with obstacles that you can and the work was done by Daniel Chevat, who was a doctoral student at the time, and subjects were taught to walk around and point, so detect the obstacles, and also avoid. So that was the task. And we see here one of the subjects doing this, so pointing and negotiating a path around the obstacles. Interestingly enough, again, for the detection, if we look at blind subjects and control subjects, they do the task, both of them very well. Larger objects are detected better than small objects, as you can see here, and the step around are detected much better than the step over. And for the avoidance, we get the same thing between large and small obstacles, and also if you have to go around a pass and avoid it or step over. So non-blinds, blindfolded, can really do the task. So this is a movie of one of our subjects. Points to the stimulus on the ground. And then it clears. And goes around and continues to do the obstacle course. question, how do they do it? What parts of the brain are taking part of this? So we built a software. Again, would be much easier if you could try to understand it, but we had three types of routes. One was straight line, was the control where the subject could move himself. So the subject would be here. As you can imagine, the pass on the tongue. The subject is here and the hand is here. And the subject clicks and moves around on this route, which is this hand. We have the second route, which is like that. We try to use something simple that we can do in a scanner, because the subject will move into the scanner. Those are the data for the learning of the days, and we see that those groups learn the task very well. If you come in the last day, they can read even before draw, because we ask them to draw the song. And the post-group did very well. There are two lines to control, so we have for route 1 and for route 2. And then we had the recognition task where they had to tell if the routes were same or different. And we see also here that they do it very well in terms of recognition. The 5, they were put in the scanner. And in the scanner, the task was passive. And when they were learning, they were doing it themselves. Now, in the scanner, they were presented the routes on the tongue, and they had to click on the same different type of task. And the flat maps of the cortex indicate, again, the recruitment of the visual cortex, lateral occipital, and interestingly, as expected, the parahippocampal gyrus, who in the controls blindfolded, they don't show the same. So we decided to take a group of subjects and give them the same task in full vision. So this time they were seeing what they were doing, and this is the result. Of course, the activations are much larger in the non-direct full vision, the visual cortex, and they recruited also the parahippocampal gyrus. Basically, these sort of activations that you see here are similar to the ones we see in the blind, but of course, actually a much lower level of activation. And if you do a contraction analysis to compare what are the areas that are common to blind and to the same controls, and you find again the visual cortex that is always recruited in the congenital blind and the hippocampal, the parahippocampal formation. Well, I mean, all these things are nice, but the blind brain being determined, if we refer ourselves to the first slide I presented to you, has undergone these plastic changes since birth, because there was no visual information coming. And I remind you that during the stages of development, you have a first phase that you have exuberance, lots of cells are there. And then you go through a phase of elimination, which is the apoptotic phase. And the third phase is stabilization. During the phase of elimination is fight for space. And during that phase, one army is missing, a huge one. It's a visual army because it's people didn't have any eyes to start with, so the retina was extensively damaged. And as I will show you, the optic nerves are basically gone. So there was nothing feeding the system, which left all the other armies, namely auditory, somatosensory, etc., to invade these territories that were occupied or supposed to be occupied by the visual system. So the idea, and I like very much, is really this invasion of territory extension of the motor. So we use these techniques, for example, for VBF, lower occipital cortex, when we can look at the metabolism of the brain and also measuring the cortical thickness. And this is what you get when you do that. And we see that, for example, the blinds in various visual areas, there is a reduction of the volume, which might be expected because they were not nourished by visual inputs. And we see also that the white matter undergoes also changes that I will show later. So if we summarize the visual system of a blind, the optic nerves are atrophied, the chiasm is atrophied, the pulvina of the thalamus is atrophied, the lateral geniculate, of course, is atrophied. We find out atrophy of the optic radiations, but they are still there. And we see that the primary visual cortex is reduced for about 20 to 25% in volume. But 25% of a big size part of the cortex is not that much because you still have 3/4 of the head cortex to do something with. It gives you a nice territory, so to speak. Other alterations that we found with the posterior part of the hippocampus, the posterior callosum also was affected. And then we looked at metabolism at rest of the brain, and without spending too much time, the back of the brain is highly metabolic, as you can see here. So there is an increased metabolism of the visual cortex at rest that you can see in this section here, compared to normal controls, where at rest the metabolism is generally distributed, and in the blind you find this big activation on this side. If you inflate the cortices to get rid of the short side again, then you will see that in the blind the visual cortex seems to be also very active compared to the sighted. So there is a supra-metabolic activity. And if one looks at the cortical thickness, one sees also, by comparing the blinds with the non-blind, that the occipital cortex, we see here, is thicker. So we have there's several conclusions that we can have here. Decrease of the gray matter. We have an increase in cortical thickness. The resting state is higher. And as I showed you with the diffuser imaging, the visual cortex can still maintain connections, and above all the occipital connections. We didn't find any new pathways. As I showed you, like for example, in the hamsters in these animal models, that aberrant ectopic projections just came in during. And as I said before, the argumentation now is maybe that this projection have increased with use in the but we don't have any evidence at this stage for research still. Now, what do they feel, these blind people? And I'm sure you all have read this saying by William James that if we splice the nerves of the excitation of the ear fed the brain center concept we see, and vice versa, we would hear the lightning and see this. So what did the brain do? And we did this very, very interesting story, I think, because it was so counterintuitive. It was so counterintuitive at the time. We used single pulse TMS of the occipital cortex, and we knew that in normal subject you would get phosphenes. And the idea was how to explain to a person who has never seen anything what a phosphene is. So what we did in the first round, we took normal subjects. We stimulated the occipital cortex. They pointed toward the phosphenes when they saw one. And then we gave them a map of the visual field and we asked them to draw them. So these are the actual phosphene that people, that's these three subjects were seeing. And you can see it's very different. People see shapes and weird things and lights. In the blind, the first, we just told them that we are going to stimulate their visual cortex, and if anything happens to tell us. And the first stimulation that we gave the visual cortex, the blind subject says, I didn't see anything. Okay. But I felt a tingling sensation on the tongue. And then I was explaining it, really? And then we said where. So they pointed at the tongue and we gave them a 3D tongue where they could also point that we could redo this thing. So Ron and myself are extremely surprised of this, and we continue to stimulate the cortex at different places, and you see where the tongue is, and the black part is the area where the paresthesia was reported. And you see for these subjects that the sonotopia of the tongue was displaced onto the visual cortex. And if you redo that towards you still have the effect. Again, I said it before, I say it again. It's all training dependent. These people are very good at using their tongue. If they are very good at using their tongue and they can do this, they're even better at reading Braille because they have read Braille on their lives, so to speak. So we decided to do the same study with TMS and just ask them if they felt anything. And this is what you get. The paresthesia, they start reporting these tingling sensations in the finger in a task, and they had to read Braille. And then we TMS the occipital cortex, and each time we TMS the cortex, we got sensation into the fingers. And then the somatotopia of the fingers also displaced towards the back of the brain, to the occipital cortex. So, conclusions, and I'm done. With sufficient training, again, training dependent, they learn to use the tongue to perform simple rotation that takes motion in motion direction. If we do the TMX, we produce paresthesia on the tongue and on the finger, suggesting a somatotopia of the visual cortex. This is more like supporting the difference hypothesis, that the brain has taken over the somatosensor has taken over the visual cortex. And of course, navigation is you can use it by using this protoxical pathway that I said before. There are big changes in the connectivity of the blind's brain. And what are the advantages of this tongue display unit compared to brain? Well, one might say it's hands-free. So that the visual information from stimuli outside the egocentric space can allow navigation, and just the talk before has been discussed. And I would like to acknowledge all these people and overall dedicate this talk to Paul Bakirita. It's a very unfortunate picture, actually, because Paul was in Montreal at a symposium with Ron Cooper and myself, and it was just a few months before he passed away. That was the last time, actually, that I've seen Paul in Montreal, because a few months later. Thank you very much for your attention.
Thank you very much for inviting me. enormously beautiful work. However, not being knowledgeable in neuroimagery, I find it very difficult to make really critical remarks. And I actually went around asking people what they thought I should comment on before the talk, and I didn't find really much. So I actually asked him what he thought I should say. He gave me a few suggestions. He said I could ask about possible explanations of the morphological and methodological changes in cognitive people. He said another thing I could ask was about the practical applications of using the Ptito device in the real world. I also asked Ptito what I might say, and he said, since I'm a sensory motor person, I could ask about the involvement of motor cortex in these changes. And I also had another idea myself, which was to ask about the relation between what Maurice has found and Ahmed's finding that visual cortex can actually be invaded by memory mechanisms in cognitively blind people. We'll talk about that. But if Maurice wants to address these questions, he can after my intervention. And after much thought, however, I came to the conclusion that I would like to share with you some more deep philosophical confusions that I have about this kind of stuff. So, when I first came upon the experiments on the rewired ferrets and later the rewired hamsters of Maurice, I was thrilled because, of course, the work shows something very profound. It shows that it's wrong to think that specific brain mechanisms are dedicated to generating specific sensations. It was compatible with the idea of the neural correlate of consciousness is what Gilbert Ryle has called a category error, a category mistake. That is to say, it's a mistake to think that one can look in the brain for something that generates the sensation of seeing. The reason, of course, it's a category mistake, I think, is that the reason it's best to think of it as a category mistake is because if you were to say that cortical mechanisms generated sensations, then you would be back into an infinite regress of additional questions as to what it is about those particular cortical mechanisms that generates those particular sensations. And you can always ask, why is it that way rather than this way? Whatever response a neurophysiologist will give you. So it seems to be a logical problem which is solved by taking the idea that this is just the wrong way of thinking about sensations. And another thing that thrilled me about these results was that it was compatible with my sensory motor theory, according to which the particular quality of experience resides in the different modes of interaction that we have with the world when we perceive. So what makes something visual rather than auditory is nothing to do, or little to do, with cortical activation, but it's to do with the fact that when you're seeing, you're interacting with the world in a visual kind of way. For example, when you blink, the information coming into your brain changes dramatically. When you move your eyes, there are certain other kinds of laws that describe those changes. And that's what the sensation of seeing is. It is all those things that you know will happen when you move around in a visual type exploratory fashion. So the results of Maurice and other rewired animal type results are quite compatible with that idea. However, an issue now arises, and it's the issue of cortical plasticity. The interpretation generally given to the kind of results that Maurice is talking about, and indeed the interpretation given generally to sensory substitution results, is in terms of cortical plasticity. It's said that through cortical plasticity, brain areas previously involved in generating vision now end up generating audition or some other combination. Now, doesn't this fall back into the same category error again? doesn't it suggest that the lesson of the category mistake has not been learned? People are surreptitiously reintroducing the idea that cortex does generate sensations after all. It just can rewire itself to generate different sensations depending on training and sensory substitution processes. So what is the alternative? Again, I think the alternative is to adopt sensory motor type approach and to suggest that what makes visual sensations visual is not the cortical information, but it's the mode of interaction. The reason why TMS to the visual cortex in Maurice's blind subjects generates tongue sensations is that what we mean by a tongue sensation is interacting with the world using the tongue. And since these subjects have been trained to do that, and they use the visual cortex to do that interaction, then when you excite the visual cortex with the TMS, you feel tongue-type sensations. The reason sensory substitution works, or at least works partially, is that some of the laws of sensory motor interaction in one sensory modality are captured and recreated using a different modality. But notice two things. The mechanism that is creating these changes is not cortical plasticity. It's just a fact about the interaction. You don't need any adaptation under my approach. What I'm saying is that it suffices for the interaction itself involved to obey the correct laws, and you will, by definition, have the sensation. Now, this seems very odd. Am I saying that if auditory cortex has only just now freshly been connected to visual type sensory motor contingencies, you would still see without any training, no cortical plasticity? So to understand the claim I'm making, take the analogy of a physical skill, like, say, skiing or whistling. When you put on your skis for the first time and start sliding down the slope, are you skiing? No, sort of. You are skiing, sort of. And the sensation you have is a start towards the feeling of seeing. And gradually, as you get more adept at this, your brain adapts and it becomes able to predict better what will happen when you change your weight or when you go over a distance. Your brain gets tuned to these sensory motor contingencies involved in skiing, and you can predict better and better the changes that will occur. So the idea is that as soon as you interact with the world in a way that corresponds to the visual way, then by definition you are seeing insofar as you are interacting in the visual way. But of course, since the brain is involved in this interaction, and since it's not adapted and isn't used to predicting what's going to happen at this time, what you will be doing at that time will only be sort of seeing. Okay? It will adapt better and better. You learn to see better and better as you go about doing it. But all the time, you are still seeing, sort of. So the role of the brain in this is to capture the contingencies, to better master them. As it does this, the sensations become more tuned to the visual mode of interaction, become more variegated, more like what normal people call vision, normal skiers call skiing. Now, doing this kind of learning, I would say, is the everyday fodder of the brain. I don't think I want to call this cortical plasticity. It's just normal learning. Other examples are extending my body when I drive. When I park my car, I feel the curve at the back of my car, on my car, as though it was part of my body. And this happens within minutes of me getting into the car. When I write with a pen, I can feel the roughness or smoothness of the paper at the end of my pen, extending the body within seconds of holding the pen. Is this cortical plasticity? No, it's just what sensation is. Sensation is not having a cortical activity in the brain. Sensation is the no adaptation, no possible rewiring. Iriki and his macaque studies has shown, of course, that the cortical fields of the fingers extend to the end of the rings that the monkey uses to bring in distant objects. So things are happening, but it's a mistake to think that those things that are happening are generating the sensations involved in sensation. And I think it's a mistake in some sense. Well, this is why I say it's a bit confusing to talk about this as cortical plasticity. It's just the everyday learning that I think the brain is doing. So that was maybe one of my points. And the second point concerning the limitations of sensory substitution systems. Fiona said in her talk, brilliant talk yesterday, that there might be limitations of possible sensory substitution mechanisms, and that they might be limited to what she called common sensibles, and that proper sensibles could not be subject to sensory substitution. She said that this might be because if we were to provide sensations that have no link to other sensations to the brain, how would it know what the quality of those sensations should be? So how could you ever substitute something like color, which apparently has no spatial aspect, for example? Now, perhaps this is another instance of the category mistake. It's a remnant. It surreptitiously presumes that sensations are produced in the brain. Under my sensory motor approach, even the so-called proper sensibles are merely ways of interacting with the world, so we should be able to approximate them to a certain extent using sensory substitution devices. Jonathan Cohen suggested that another limitation of sensory substitution devices might be what he called emergent features, since these are, so to speak, created by higher cognitive mechanisms that are not to be actually found in the input stream. So we couldn't expect a sensory substitution device to transmit them. I think this is perfectly reasonable, but I wonder whether the whole motivation underlying Jonathan Cohen's question was not again surreptitiously biased by a kind of secret hope that it might somehow actually one day really be possible to create complete, in some sense, sensory substitution. Under my view, by the very definition of what sensations are, this must be impossible, because if what sensation is interacting with the world, as soon as you change the mode of interaction using a sensory substitution device, you change the sensation. The perceived quality must necessarily be different, since it's composed of the sum total of all the things you do when you interact in that particular way. Sensory substitution can be partial, though, and you can get certain kinds of information from the environment using substitution devices. But it's ridiculous to think that one should be able to really totally see through a TVSS. So my approach would be what you might call deflationary about sensations. The questions of whether you really do see with a TVSS is a kind of remnant of the neural correlative consciousness category mistake. Taking the sensory motor approach, it makes it a silly question to ask. It's sort of like asking whether you're really skiing when you're skiing on one of those game parlors ski simulators. It's a silly question, like the question of spectrum inversion. Could you envisage, for example, that squishing a sponge, feeling it seed under the pressure of your fingers, would give you a feeling of being hard as a stone, despite the fact that when you press it, squishes? No, because pressing a sponge and obeying the laws of softness defines what it is to be soft. And it's ridiculous to think that you could be squishing and obeying the laws of softness and then somehow feel it as being hard. So in conclusion, I apologize to Maurice for not commenting directly on his talk. Apart from the more concrete questions that I posed at the beginning, the overall question I have is he and are you happy with using the notion of cortical plasticity in describing these phenomena? Do you really think that through the rewiring of the cortex, new areas of the cortex are generating new sensations? Thank you.
love to see Kevin do philosophy, but I'm going to miss my chance to comment and ask Maurice a question, if I may. An observation and then a question. One thing is, I wondered if you realised that you were also using, for the mapping on the tongue, you were using natural cross-modal correspondences, because people typically experience sweetness at the front of the tongue, bitterness at the back. And I'd like to know whether this is true that cortically blind, but in normal subjects, sweet is associated with high musical notes and bitterness with low notes. So it might be that just having done the mapping the way you did, perhaps even implicitly you knew to go for front of the tongue and back of the tongue. If it was reversed, I wonder if it would be longer and more counterintuitive. That's one thing. Second thing was, I was wondering if there are residual effects of using the tongue so that when they have, say, a glass of champagne, or when they have something that's actually provoking different places of the tongue, they get something like spatial illusions or hallucinations just because you're using the tongue.
Okay. There are many things in your question. Usually the blinds, as seeing people, when we do this test, they use the tip of the tongue. So it's an anterior third that is mostly used. Second, in the beginning, they do have fierce sort of taste that wanes after a while when you do train. In the studies we did on taste, and we are pursuing now, this is probably the only sense where the blinds are much worse than the seeing people. For smell, they do better. They, so to speak, smell better. We know from audition that they can detect and localize better. And for the sense of touch, we know also that they do better. Temperature discrimination, touching things like that. For taste, they don't. They really do worse than seeing in a way. So the idea or the explanation maybe for this would be that their repertoire of foods is so limited. When we eat and we want to try something, we see it, and if it looks palatable, we sort of it looks good, right? They don't, and they are very scared in a way of eating foods that they don't see, of course, and they tell us when we go out, we take them out, and they ask what's on the menu, and we tell them they will stick to something they know. If they eat hamburgers or chips and things like that, or we say, why don't you try this kind of thing? Oh, is it good? Well, try it. So they will rely on you to try it out. So, we were, besides the first parts of this taste that wanes with time, we don't think there is an effect really on the taste like a cross-modal kind of things. But it might be, I don't know.
It's a last question that arose first for Kevin, but I can reword it so that it's addressed to the two of you. So you just said that there's a priori no limitations of sensory substitution, and it's not limited to spatial senses, but in fact it's the system that works the best, the system that are devoted to spatial senses, and there are really nice work of Brandon-Yancey that try and substitute for pain, and the result is that it doesn't work. Either the signal is by itself painful, and then the subject interprets it as a punishment and no longer use the device, or the signal is not painful and participants ignore the signal and keep on being hurt. So, would you have a sensory motor theory of pain that would account for the fact that there is no good substitution of pain, or would a sensory motor theory could help understanding what could be a code to substitute for pain? And in extension, is there something about cortical plasticity that explains that we substitute well between spatial senses and not between senses that are less spatial or pain or taste?
Well, with regard to pain, I think it's very interesting. I think what characterizes pain, from my point of view, one thing that really characterizes pain is the extreme attentional grabbiness of pain. And my claim would be that you should be able to create artificial pains if you could recreate that same grabbiness. Essentially, if we could create stimuli which stop you from thinking about what you're thinking about and incontrovertibly direct your attention to something. And if you interpreted those stimuli as being bad for you and socially bad for you, then I think that amounts to and that constitutes it being painful. So I do think it should be possible to create artificially painful stimuli. The problem with the experiments you were talking about was that the person would just tear off the device which attracted the person's attention. So
there was two things. First, they try and derive the stimuli in another location on the body, but keeping it painful. And it wasn't interpreted as there's a harmful signal there. It was interpreted as a punishment. So they didn't substitute for pain via a painful stimulation. So then they tried to vary the signal to find a code to say it's painful. But in all their attempts, they didn't succeed either.
Yeah. Well, that's only one experiment. I think pain is very interesting, and I do think, according to my theory anyway, it should be possible to, if you could capture attention in the appropriate way.
It's captured attentionally anyway. You cannot do more than having something painful elsewhere.
Yeah, but it's in the wrong place, isn't it?
Hi, this is a question for Kevin and Reagan. I guess I was wondering what you think the best empirical evidence for the sensory motor contingency theory is. Going back to your 2001 paper and the noise book, we had a lot of cases were put forward. So it was Richard Hell's experiments on kids and work on adaptation to optical rearrangement devices and work on TVSS. And critics, ranging from people like Ned Block to Jesse Prince, have gone through those examples and have offered alternative interpretations that to many people are pretty persuasive. So I guess I'm wondering what you think is the best empirical evidence for the sensory motor contingency theory. Which cases would you appeal to?
So I think like all science, once you've got a phenomenon, you can reinterpret it in many different ways. And so what makes a theory interesting and useful and perhaps true, almost true, is the fact that it makes new predictions which are counterintuitive. And in fact, I attribute the interest of sensory substitution that has been developing over the last years to this kind of sensory motor approach. It's because our theory suggested that sensory substitution wasn't just a dream, and that it might be possible, that the whole paradigm of sensory substitution has become more alive. So that's one thing. I agree it's not proof, and you can always reinterpret it different ways. Something that I'm particularly proud of is my predictions about color. I don't know if you've read the color papers, where I'm able to show completely from first principles why red is a color that is named by cultures throughout the world, whereas pink is not. Red, green, yellow, and blue are primary colors, and I can predict why that is from a study of the sensory motor laws that describe the changes in light when you move some red paper around. So that's what I think is perhaps one of the best proofs, if one can talk about proofs.
So I've got a question for each of you. So for Maurice, my question is to do with the TMS over the occipital lobe. Why is it that sometimes when you stimulate the occipital lobes you get the tongue, and at other times you get the hand? Is it kind of type specific?
Yes, it is that specific. In the tongue, the subjects they were trained on doing a task, and then they were TMS, and then they report sensations. Same where after having done a braille task of reading, they were stimulated or they were seeking good.
So this means it's kind of like a short-lived echo rather than an organisation.
But if you redo the same simulation 2 months later, you still have the paresthesia. They're still there.
Okay, so if you can read braille and you use your tongue to this, why do you get it going to one and not the other?
Why sometimes it is the tongue and why sometimes the fingers, depends what organ you use to do the task.
Okay, right. So they're actually doing a task once. Okay. And with Kevin, it's also related to braille. So how does braille reading kind of obey the sensorimotor qualities of vision? Well, I don't misunderstand that. In order to get these kinds of things. And also there's a paper by Ortiz in +1 in which blind people are trained to make orientation discriminations with their hand using pins. And after several months it's found that about half of them report visual phenomena, including luminance. And so those who report the luminance activate their occipital cortex, and those who don't report it don't. So what is going on here? There is no sensorimotor contingency. They're just putting their hand on an array and processing orientation.
Well, I think they previously processed orientation using their hands in different ways, moving them around, and they have previously been subjected to visual stimulations which have intensity differences. The laws that they have previously sampled in that way actively are activated by the very reduced information we're getting by just keeping their hands on the stimulus.
They were late blinds. It was a mixed group, and then they divided them up. I guess most was in the beginning of this presentation. Late blinds are really something very difficult to work with, because one has to consider onset of loss of vision, and secondly, for how long they have been blind. If somebody has been blinded at the age of 4, and so already had experience for 4 years of vision, and then is about 60 years old, the brain has retransformed into a sort of blind brain. And we have a sort of evidence for this. Now, what happens with late blindness is that people who are using late blinds, it's a group. So they don't take into consideration when it happens, for how long they've been blind, and so on and so forth. That's why you have this type of...
I agree with you, but I'm not sure that the sensorimotor theory makes that distinction between early and late blindness.
Fiona. Thank you. So I have a question for both as well. So from Maurice, I had a very similar question to that Jamie just asked. So am I right in thinking that your answer to Jamie was that if you stimulate the visual cortex of someone who's blind, it depends on what task they've just done previously, what they report. So if they've just done a braille reading test, then they report sensations at the fingertips. If they've just done a TDU task, then they report sensations in the tongue. If they've just done the TDU task, then they report sensations on the tongue.
Yes. Yeah, they have done a task for some discrimination, for example, with the tongue, and then they were sort of TMS afterwards.
But it's afterwards that you do is it during the time that they're doing the task? It's
after training, not during the test. We have done some. As I showed on the experiments we have done on motion, and have shown the area MT was active. And in this case, we haven't shown it here, but we done the TMS simultaneously with the stimulus while the person was performing, and then you can interfere with the performance, because you interfere with the part of the brain area that is responsible. And have that. but after you are trained, and let's say, so to speak, your tongue is trained, and then you use TMS, and don't forget this subject has been trained for quite a while.
I see. Thank you. That's helpful. So I have a question for Kevin as well. So in response to what I was saying in my talk, you said, well, look, you think I'm wrong that if you just, you think that if you recreate the mode of interaction, then you could recreate sensations and proper sensibles because you would have the right sensory motor loop. But then in response to what, your sort of response to what Jonathan said was, yeah, but hang on a minute, because of course you couldn't really create exactly the mode of interaction because the mode of interaction is sort of very highly dependent on the sort of perceptual system that you're using. So if that's true, it looks like you think that you couldn't really recreate the mode of interaction, so we couldn't have sensory substitution for taste because unless you used the tongue, then you wouldn't have the right mode of interaction. So if that's what your position is, it looks like you think that there are limits precisely to sensory substitution.
And that's what I was saying. I'm saying there are limits, but they're not for the reasons you suggested. I think it seemed to me that the reason you were suggesting that there were limitations was something to do with brain structure or brain computation that was generating particular sensations like taste and colour, which had no common link to other modalities. And for that reason, the brain wouldn't know what link to make with what was coming in, and so it could never recreate the sensation that you were trying to get it to create, which was subreptitiously containing the idea that different brain regions do different sensation type generation.
So I didn't mean to build in that assumption to what I was saying. One way you could try and reinterpret what I was saying in your terminology and in line with your theory would just be that there's not enough information in the kind of signal you could give about taste such that you could recreate a specific enough sensory motor loop that would correspond to taste rather than to a sort of simple form of smell or to touch or to colour, or so there would be nothing sort of there would be no specific enough sensory motor loop to sort of cotton on to. That would be one way of putting it.
Well, no, I mean, Charles Spencer was saying yesterday that there's 1 bit per second of information in touch or in taste, say, which is not very much. So you should surely be able to create a simulation of the input stream to taste, which is just 1 bit per second. But the trouble is that the action component of taste, you've got to move your tongue around, and the tongue is a complicated organ. So in order to really get something that felt like taste, you'd have to have the tongue component in there. And furthermore, I think taste is connected with a whole lot of other automatic responses of the nervous system. Like if you taste something very sour, your mouth kind of buckles up, and this gives automatic responses, which you'd also have to include in the sensory motor loop, and that would be difficult. But if you could do it, if you could simulate it, well, then I think you should be able to get taste substitution.
So I think the gentleman in the back corner wants to come in on this question. Is that right? Is this a follow-up on this question?
Yes. Basically, I wonder, I totally agree with you that things are, almost everything in the world is sensory motor loops. So I wonder, and follow up on what you said, and I suspect you do agree with it, is that the system and the experience might pose some constraints, and this is because otherwise all the brain would do the same thing. So I wonder what to think about these constraints and whether it's useful to try and think about what are the constraints, why do we have these types of tasks, different tasks, and these different sensory action loop, and whether we can influence and change them and change perception at the same time.
Right, I agree with that. There are constraints, and there might actually even be computational constraints in the brain. And I think maybe visual areas and auditory areas and somatosensory areas might actually be, by evolution, pre-wired to do certain kinds of computation that are better for extracting information from those different sensory modalities. And the kind of work you are doing, Maurice is doing, is of course very interesting with regard to those questions. But I think there's a fundamental philosophical error that
questions, if I may. The first one, from an observation, when we look to the literature on blindness and cross-modal brain plasticity, we observe that the occipital cortex is recruited in great variety of situations, stimulation, task, and some do not require any training, think for instance, or some processing. I found very interesting the experiment you did with TMS. When you apply TMS in your blind volunteers, so you get sensation on their fingers or tongue, did you observe any volunteer? Did any of your volunteer report some auditory perception during the stimulation?
No, because we didn't ask. Sorry, we didn't ask. And we are wondering, which one at one point, why did the people working on the auditions substitution, they didn't, you should have some sort of auditory for sphinx, I guess, some, yeah, but we didn't ask, so I don't know.
Second question, maybe a tough one, but that's a question that I find very interesting, and I'm looking for answers also. You observe brain activity in the occipital cortex of the brain. Do you think that some part of this brain activity are related more to the task and while other part may be more related to the sensory processing? In other terms, did you already observe brain activity during a passive stimulation when your subject are doing nothing?
No. Usually it's always where they are doing something.
Yes, but did you try just passive stimulation without any orientation task?
What do you... in the motion study, we also had some incoherent motion, and we could kind of consider that as a control task. And also we found some activation area empty. But if you contrast between the coherent motion versus the incoherent motion, we will get much more activation. But we, particularly first study, that most of them being bad studies, so we didn't have much possibility to include too many control conditions. And the other problem, of course, is when you have a limited number of subjects. And when the first time you do these TDU experiments, you have genuine naive subjects. But when you have that been going on and they've been studied already in 2 or 3 studies before, they are no longer anymore naive subjects. So actually, the fact that we lose some kind of the advantage that we have at the beginning, that we have a pre and a post condition. So I think that's also maybe important to mention that the novelty of TDU, if you have to always take the same subject, you can't lose the fact that you have a pre and a post condition in general.
Yes, thank you. One of the questions that is behind this possible sensory processing is, of course, the question of where comes the non-visual input inside the occipital cortex. So does it come from cortical connection or thalamocortical connection? And remember one of your papers about olfactory processing. I think it was passive. You reported activity in the occipital cortex. And so that's one of the questions I'm pursuing also in my own work currently.
There was a detection. That was not passive. The subject were asked to identify which order was presented. So it was definitely not a passive task.
Okay, very bad. Thank you.
Okay, we've got 3 questions to try and fit in, maybe 4. So the next questioner is the gentleman in the brown jacket here.
I would hate to say the 2 good questions Kevin asked in the end of his commentary as well. So I'll just repeat them for Professor Ptito. One was, I think, is cortical plasticity just plain old learning? And the second question was, is it categoristic to think that certain cortical areas generate certain types of sensations? That is this.
Cortices are sought usually to generate a particular sensation in area V1, for example. You would have a visual sensation that will originate there. More and more we find connections going into from other sensory cortices to like the cortices between them, like recording, for example, auditory responses into the visual cortex, for example. So this is coming more and more. So it's a sort of a gray zone now onto these things. So learning, my belief is that cortical plasticity and learning are very tight because, when we learn, we create new connections. So I believe that this plastic process is going through the whole lifespan where cortical plasticity occurs continuously, in my opinion.
A question for Maurice. You very briefly mentioned intraocular stem cell implants and a couple of other things along the same line. Could you just say a word about what ways those are comparable or different from these other devices?
These techniques are highly invasive. So they require surgery and pain. I don't know if you're aware of the Tactile technique that one has to put a grid of electrodes into the striate cortex, which is neurosurgery, so to speak, where you have about, 300 to 400 electrodes implanted, and all the wires coming out from the head, and these electrodes are stimulated to create phosphines, and stimulate part of the visual field and reproduce, so to speak, some images in people who became blind. So they had visual repertoire before, which is really key for these kind of things, because as I said before, for blindness, congenital blindness, people will have no visual repertoire cannot form images because they don't have it, so to speak. So this technique is probably one of the worst, this Tactile technique, because you can imagine something was wrong with connections or with the electrodes, and you need another surgery to remove or replace, etc. Actually, I'm extremely surprised that the double clinic is still there, and they have it in Portugal, I think, where there is a clinic. The retinal implant is quite interesting. If you have retinal implants still there and stimulated, the information can, of course, go to the visual cortex and again sort of reproduce images by stimulation in a case where you have lost the vision but we had it before. And more interesting, and I think would be the future, is the stem cells. The data so far is quite encouraging because if you have a crushed optic nerve and stem cells are implanted in this area, the nerves will regrow. So there is, we have two cases in human, one for the regeneration of the auditory nerve by the injection of stem cells that worked pretty well. And I was actually surprised to I've seen this patient, and they re-established the hearing in the patient. So I think these techniques, to a certain extent, the reason why they are, besides just stem cells, they're all so invasive, and we don't know to a certain extent, what kind of things are behind. I'm really more in the stem cells because I've seen some of this stuff that they're really exciting.
So are these attempts to restore vision, or is there something
that is attempts to restore vision? Yeah. Now, the other system, and it brings again the idea about using these substitution devices. I mean, as a tool for research, I think it's amazing. I really love this thing we're doing with this TDU, and it allows us to tap on brain functioning, brain connectivity, and all kinds of things like that. But the use for the blind is really, very limited. I wouldn't put the TDU on the blind and send him outside in the street. He would be dead in 2 minutes. Right? Even us seeing people walking in London, you have to rewatch it not to be killed. But in a setting, in a laboratory setting, everything is controlled. All the work we've done, for example, with these subjects, this blind subject, the system works on optimal intensity in terms of contrast. Like, for example, black or white. If I put a black bar, as I've shown you, on the white background, the subject will move his head with the camera. Once it would catch, it enter the black part, the signal would come to the camera saying, there is something black there, or there is an object there. And you've seen also that in the corridor, all the obstacles are black, and the walls are white. So, to a certain extent, we facilitated that, but in the real life, it's not black and white. So they would have no problem of doing tones of grays and things like that, and color, as we discussed before. 3D, they've tried now to have 2 cameras that are doing binocular vision, and it's quite complicated for simple things. I think moving into the corridor, I was very amazed for navigation, and we saw how they could point, and also they could step over the obstacles, and they do it so well.
Apologies to those who've been waiting to ask questions. Hopefully you'll find an opportunity to do so later in the day. And thank you very much to both our speakers.